This has been quite the journey through metabolism. There's been a lot of enzymes, pathways, and disorders that you'll need to know for the test. Make sure that you're comfortable with this material so you don't miss an easy test question. Let's do some practice questions now to demonstrate how biochemistry metabolism questions can appear on the test day. Just remember though, I chose the hard questions that'll really make you think. Not all questions on the USMLE will be this difficult. So let's go ahead and begin. In a healthy individual, fasting leads to increased lipolysis, increased accumulation of fatty acids, and increased production of acetyl-CoA. Acetyl-CoA is an allosteric activator of an important mitochondrial enzyme that catalyzes a rate-limiting step in gluconeogenesis. What is the product of this reaction catalyzed by the enzyme? Is it A, fructose 1,6-bisphosphate, B, glucose, C, glucose 6-phosphate, D, oxaloacetate, E, PEP, or F, pyruvate? I'll give you a second to think about it. Ready for the answer? Well, if you chose D, oxaloacetate, you would be correct. And let's take a look at why this is the case. The question is simply asking two things. How is gluconeogenesis regulated? And then B, what is the product of this step? How is gluconeogenesis regulated, and what is the product of this step? Remember from our conversation on fatty acid metabolism that fatty acids get converted into acetyl-CoA molecules. Gluconeogenesis occurs in the liver when the body wants to create glucose molecules like during hypoglycemic fasting states. To make this glucose, the body is going to need to reverse the glycolysis pathway. Many of the enzymes in the glycolysis pathway are reversible, except for these three irreversible steps. Thankfully, we have four unique enzymes in gluconeogenesis that'll bypass these irreversible steps of glycolysis. A molecule of pyruvate, down here, is first converted to oxaloacetate by the enzyme pyruvate carboxylase. This step is allosterically activated by high acetyl-CoA levels. So when there are high levels of acetyl-CoA, pyruvate carboxylase will start converting pyruvate into oxaloacetate, and gluconeogenesis can then occur. A 55-year-old man with recurrent arthritis of both knees presents to his primary care physician for a routine checkup. Results of urinalysis are negative, but the nurse reports that the urine sample turned dark upon standing. Levels of what substance will be most notably elevated in the urine sample from this patient? Is it A, branched amino acids, B, cysteine, C, homocysteine, D, homogentisic acid, or E, phenylalanine? The answer here is going to be D, homogentisic acid. This question gives some good clues to as what the patient has. First, the question says that a patient has recurrent arthritis. Also, his urine turns black upon standing. These two findings together suggest alkaptonuria, which is the result of defective tyrosine breakdown. Bad tyrosine amino acid breakdown. So the enzyme that is going to be missing here is going to be homogentisic oxidase. And this is going to convert our homogentisic acid to a mallyl acetoacetic acid. Therefore, high levels of homogentisic acid are going to lead to black urine and arthritic change because we're not able to proceed with our tyrosine breakdown. A 6-month-old boy is brought to the physician because he seems smaller than other kids his age and occasionally vomits when given bottled milk feedings. He was born at home and did not undergo newborn screening. Ooh, that'll make you cringe. He did not undergo newborn screening. On physical exam, the patient is found to have jaundice, bilateral clouding of the eye lenses, and hepatosplenomegaly. The patient also shows signs of developmental delay. Which of the following enzymes is likely to be defective in this condition? Is it A, alpha-1 hydroxylase, B, aldolase B, C, galactose 1-phosphate uridyl transferase, D, iduronate sulfatase, or E, transmembrane copper transporting ATPase? Think you know the answer? The defective enzyme here is C, galactose 1-phosphate uridyl transferase. This question prompt gives us all the information we need to figure this one out. So we have a 6-month-old boy. He vomits after feedings. He's smaller than other kids for his age. Notice that we have clouding of the lens here, and also hepatomegaly. So here we have this kid that has a lot of buzzwords associated with him. With all this in mind, hopefully you're thinking of classic galactosemia, which is going to be an autosomal recessive disease that's caused by a deficiency of our galactose-1 phosphate uridyl transferase enzyme. Remember that there are two galactose metabolic disorders. The milder form is going to be the galactokinase deficiency, and it may present with infantile cataracts, but it usually doesn't have the developmental delay because of its benign nature. Therefore, I'm more inclined to think that this is going to be more classic galactosemia. Remember that our galactose-1 phosphate and galactitol will accumulate in organs, and also galactitol. So these will accumulate in the organs, causing damage. Notice how the question said that vomiting occurred primarily after feedings. Milk has lactose, which is a disaccharide that gets broken down into both galactose and glucose. Therefore, the treatment for this little tyke is a lactose and a galactose-free diet. So remember that we're missing an enzyme, so we're able to phosphorylate our galactose, but we're not able to further break it down. So we have all this phosphorylated galactose that gets trapped inside of cells. Here's another scenario. A 2-year-old boy presents to his pediatrician with hepatosplenomegaly, failure to thrive, and progressive central nervous system deterioration. A liver biopsy shows that hepatocytes and Kupffer cells have a foamy, vacuolated appearance. The pediatrician suspects that this boy will die by the age of 3. Which of the following is the function of the metabolic enzyme deficient in this patient? I'll give you a second to read through the answer options here. This is a tough question that expects you to figure out the deficient enzyme, but also know what reaction it undergoes. Let's figure out what disease this question is referring to. So looking at our answer options, we see 5 possible lysosomal storage diseases. In our question prompt, we see hepatosplenomegaly, which immediately rules out a few of our possible lysosomal storage diseases. Do you remember which of those lysosomal storage diseases has hepatosplenomegaly? Well, the 3 are going to be Gaucher's disease, Niemann-Pick, and also Hurler syndrome. So we have narrowed down all of the lysosomal storage diseases just to 3 possible choices already. The next major clue for me would be the description of the liver biopsy. Here we're going to have hepatocytes and Kupffer cells that have a foamy, vacuolated appearance. So I'm just going to underline that because that'll give us a clue. It kind of sounds like they're trying to describe a foam cell, right? In Niemann-Pick disease, foam cells are a key buzzword. Okay, so we've now determined that this boy has Niemann-Pick disease. That's only half of the battle though. We now need to know what metabolic enzyme is missing. Do you remember how we can remember the Niemann-Pick enzyme? Well, you pick your nose with your finger. So sphinger. Sphingomyelinase is missing, which is going to convert our sphingomyelin to cerebroside. So our answer is going to be E. A sphingomyelinase deficiency is going to cause sphingomyelin to accumulate, so option E is our correct answer here. What other buzzwords might you see with Niemann-Pick disease? Well, if I heard foam cells, Ashkenazi Jew, cherry red spot, or hepatosplenomegaly, I'm really thinking about picking my nose with my finger. The lysosomal storage diseases are going to be a hot topic on boards. Just make sure you know them in and out. A 30-year-old previously healthy man is seen in the clinic for several weeks following up on a bout of pancreatitis. The patient does not drink alcohol and they have no history of gallstones, they're not obese, and they're not diabetic. In clinic, blood is drawn and it's found to be milky white in color. Which of the following likely explains the pathophysiology of his underlying condition? I'll let you read through the answer options here before telling you what it is. Go ahead and pause the video if you need more time, but I'm about to tell what the answer is. The correct answer here is going to be option C, a lipoprotein lipase deficiency. This patient most likely has type 1 dyslipidemia, also termed hyperchylomicronemia. In this disease, there's an excess amount of chylomicrons in the bloodstream. This is what causes that milky white appearance to the blood, due to the high triglyceride level. Also, hyperchylomicronemia can cause pancreatitis and hepatosplenomegaly. Do you remember what the two causes for hyperchylomicronemia are? Well, it can be caused by a deficiency in the lipoprotein lipase enzyme, which is typically found on capillary walls. It helps digest chylomicrons and cleaves off triglycerides into fatty acids. Lipoprotein lipase also requires the cofactor apolipoprotein C2, which is found on chylomicrons. If there's a decrease in the apolipoprotein C2, there could also be this hyperchylomicronemia. If you were to run a lipid panel on this patient, you would suspect that his triglyceride levels would be elevated off the charts, often in the thousands range. However, his LDL and his HDL levels should be pretty normal. Whew. And that concludes the biochemistry section. I know we covered a lot of material, but I want to congratulate every single one of you out there for putting in the time and the effort to do well on your USMLE Step 1. All of this effort will be worth it in the end, I promise you that. I'm Tyler Foster, and I hope you'll join me again in the pharmacology section.